I'm here to talk about when we talk in research, we talk about bench to bedside. And bedside is important, because what we're doing is translating this fantastic work you've heard about from genetics and from, TMS, and then we have to apply it to people, and people are strange. They're not mice. We can't put them in a maze. We can't, take out their brains and look at them under a microscope. So we have to do clinical trials. And when Kayleen came to me and said, look, we found these really interesting results from our mouse models, we want to scale this up into humans, we want to look at TMS in humans with MS to see whether we can add myelin to the human system. It really was a difficult question, because how do you do that? Firstly, we heard the problem of downscaling magnetic stimulators to mouse size. Then we have to take what was learnt in mice and upscale it to human size. And that included getting the coils right, getting the intensity correct, so that we could actually do a clinical trial. And what we did was, firstly, we developed a suite of clinical trials, and I'll explain why we have to do this, because clinical trials, you can't just go out and say, let's just recruit, X number of people, bring them together, and we will zap them all and we'll find the answer. It doesn't quite work like that. So what TAURUS 2 is a phase 2 trial. It's a phase 2 trial of transcranial magnetic stimulation to improve myelination in people with multiple sclerosis. It's a follow-on to what was called the TAURUS 1 trial, which some of the people in this room participated in. Now, what is a phase 2 trial, and why do we talk about these phases of clinical trials? Well, it's really important because you have to go through this process. Phase 1 trials are basically done to show that your intervention is safe in humans. We don't know that. We thought, gosh, this has been done for all sorts of things, but never in the way that we developed in mice and then into the human trial. We don't know that giving people with MS transcranial magnetic stimulation may not have had an unexpected and adverse event. So we have to do a phase 1 safety trial. That trial doesn't tell you whether the treatment works. It just tells you that we can do it, it's safe, and it's tolerable, and people don't throw their arms up and run out of the room and say, there's no way I'm ever having that done to my head again. So that's what we do. That's what a phase 1 trial does. A phase 2 trial is about, does this treatment actually work? Does it really do what it's meant to do? And usually you need more people in a phase 2 trial, and you need to, and again, it has to be placebo controlled, because the placebo effect is incredibly powerful. If I talk convincingly enough to people, I can potentially make them remyelinate their own brain just by, power of thought. And then that introduces bias into a study. So we have to have a system where the person who's getting the treatment doesn't know which treatment they're getting, and the person who's looking at the data doesn't know which treatment that person had. And that's called blinding, and that's an incredibly important part of a trial. And then if a phase 2 trial is successful, then you move on to a phase 3 trial, which tells you, is the treatment you've developed better than the treatment we had before? Now, in remyelination, we don't have any prior treatments. There's not been any successful remyelination trials ever. There's been some suggestions in some trials, but there's nothing that's been shown to actually convincingly add significant amounts of myelin to the human MS brain. So, what did we learn from TAURUS 1? Well, we did learn that it was safe. We enrolled 20 people. We gave 2/3 of them actual magnetic stimulation. We gave 1/3 of them a sort of sham, and that's a long story about what really happened there. But we knew that it was well tolerated, and we knew that we could blind people. People didn't know whether they were having the treatment or not, which is really important, because if you think you're not having the treatment, it's like putting your mice in a box. You think, why am I here? What's going on? It's not good for you. Whereas if you think you're getting the treatment, it's all like, wow, this is great. I'm having this new treatment, and it can introduce bias. So we would ask people at regular intervals, which treatment do you think you're having? And people would fluctuate wildly. They think, oh, I'm definitely having it. No, I'm definitely not having it. I'm definitely having it, which is what we wanted, because it removed that bias. It also told us it was tolerable and it was feasible, that people would come in, and have treatments. Excuse me, someone's phoning me. I'm sorry. So the people come in and have their treatments every week, every day for weekdays for 4 weeks, and that they found that it wasn't too intrusive, and that it was it was something that was entirely feasible. Well, so I'm not. Oops. So what we learn from TAURUS, we didn't learn. We don't know whether the treatment worked, basically. There was some suggestion from the first part of the trial, particularly on the MRI scans, which Jenny mentioned, that the MRIs are getting so much better. And with our collaborators at the Sydney Neuroimaging Assessment Centre, led by Michael Barnett, they developed special techniques, or application of special techniques in MRI scanners to allow us to look at myelin maps or in the human brain. And there was a suggestion, and it was only a suggestion, that we actually had made a difference, that the people who had the treatment maintained their level of myelin, where the people who did not have the treatment appeared to lose a little bit of myelin. But none of this could ever possibly reach statistical significance. Hence, we developed TAURUS 2, which is basically a much larger trial of transcranial magnetic stimulation in people with MS. It's still, the aim is to enrol people living with MS from 6 centres around Australia, and it's got too big for us to do it in Hobart alone. We need to be able to enrol people from multiple different centres. That's very frustrating. It's a randomised, sham-controlled, blinded, parallel group safety and preliminary efficacy trial. And what that basically means is, again, we're going to enrol people with MS. We're going to, 2/3 of them will get active treatment, 1/3 will get sham treatment. Again, it's blinded, so the person who's getting the treatment doesn't know what they're getting, whereas unfortunately we can't blind the operator because they've got to give the stimulation, but the people who analyse the data won't know which treatment allocation. We have got 6 sites. It's underway at the Menzies Centre in Hobart, which is the coordinating site, and we just reviewed our 11th patient yesterday, or 11th participant yesterday, which is a great result. It's also going to be, people are being enrolled at the John Hunter Hospital in Newcastle, the Alfred Health, or at Alfred Hospital in Melbourne, the Perron Institute, where Jenny works, hopefully will be starting very soon. Next week, we're going to start enrolling people. The Launceston General Hospital, they're having some trouble with getting their MRI scanner to do what it's meant to be doing, but hopefully they will be up and running. And the final centre is in the Mater Health in South Brisbane in Queensland. So we'll have 6 sites. We're aiming to recruit 108 participants, and we're hoping to complete recruitment by the first quarter of 2024. Our primary objective is, does magnetic brain stimulation improve functional outcomes, both in upper and lower limb function? We do what's called the Multiple Sclerosis Functional Composite Score, which is a measure of a 25-foot timed walk, a 9-hole peg test, which is a way of looking at dexterity of the upper limbs, and a measure of cognition called the Symbol Digit Modality Test. We want to know our secondary objectives. We want to know if that improves. Our secondary objectives are, is it safe and tolerable? Does it improve quality of life? Does it reduce anxiety and depression? Because maybe the effect we're seeing is because we're getting the same effect that you see if you use TMS for depression and anxiety. And is that what we're seeing rather than actually an improvement in MS? Does it have an effect on fatigue? Fatigue is the most intractable symptom in MS. And many people will tell you about how, and we heard it this morning, how the fatigue is indescribable, it's overwhelming. Can we affect that? Does it improve sleep quality is another thing which we're very interested in, because sleep is a fundamental property of life, or a fundamental basis of life, to be able to sleep and sleep well. And people with MS, we know from research done here at the Menzies by Laura, who's somewhere here, there she is, that shows that people with MS do have poor quality of sleep, and that sleep may be a primary symptom of MS. And we're very interested in, can we affect that important aspect with our treatment. So the tertiary objectives, which means the third level, is basically a lot of that is around the MRI. Now, the reason why these are not the primary objective is because remyelination on MRI scan is not a proven outcome in clinical trials. So we can't have it as our primary outcome. So we have to have it as one of our tertiary outcomes. And we want to really know, can we add myelin to the brains of people with MS? And we're hoping to show that by people when they enroll in the trial, they have an MRI and its particular sequences done before they start and after they finish. And we are hoping that that time period is long enough to show us that we can add myelin to the person's brain. And again, we want to know that as we see here, that our sham and our treatment are acceptable and that people can't recognise which one they're on. That's really very important for us. So what does it look like? Well, we now have these machines, which is a magnetic brain stimulating machines. They're readily available. You can go on the internet if you really want to and buy one. They cost about $70,000. We've got we have a coil, which you can see there in the middle, and we basically we give the treatment as shown here. And these two of our PhD students who have been very instrumental in the delivery of transcranial magnetic stimulation, showing how it is done. And each MBS session lasts about 15 minutes. It's generally, really very well tolerated. In our experience now, we've done a large number of these sessions now. And these are the instruments we're going to use. This is the 25-foot timed walk, the 9-hole peg test, and the simple digit modality test. These are standard MS tests, and many people who have participated in clinical trials will have used these tests. They're not, they don't involve sticking needles or pins into people, and they're done really quite quickly and quite easy to complete. But trying to show a clinical and meaningful change is one of the most important things about a clinical trial, and we're hoping that we will show some improvement on these functions. So, how to be eligible for this trial? Well, basically, we were looking for people with stable MS, and we're looking for people with a degree of MS disability. They must have something that we can measure on their expanded disability status score, but we need people to be ambulant, to have an EDSS of less than 6. Now, that's what happens in clinical trials is we have to restrict the population so we can try and get some of that homogeneity which we heard about from Jenny, because if you have such vastly different people from people, you may find that just the difference between people entering the trial prevents us finding an association with the treatment. We have age restrictions, and we really have some restrictions which are very important, that you must be able to have an MRI scan. You can't have things such as active epilepsy, active psychiatric disease, be on certain medications which affect the way that MBS works. And so we do have to be very careful about these, and you can't be pregnant, breastfeeding, or planning to become pregnant during the time of the study. So what happens if you want to participate in the trial, and we'd invite anyone who meets this criteria to consider being in this trial. People seem to quite enjoy it, actually. You undergo a screening visit, which is where you determine the eligibility. We go through our eligibility checklist and make sure there's nothing in that people don't have metal in their body, they don't have a contraindication to being on the trial. They then have daily visits Monday to Friday for 4 weeks, which is 20 sessions of MBS. They have and a 3-month follow-up visit, which is usually done remotely. MRI and questionnaires are before and after treatment, so you have to have 2 MRI scans within a 6-week period. Where are we up to? Recruitment has commenced in Hobart, and we have now enrolled 10 participants, which is fantastic. We're really very pleased with this screening, and now we know that Newcastle and the Alfred Health have undertaken and commenced screening and have started treatment in Newcastle. We've just heard that Perron is starting next week, which is just fantastic. Launceston General Hospital's still got a few MRI issues to sort out, and the Mater in Queensland is awaiting final approval. So we're hoping that we'll get all of our sites up and running very shortly. Oops. There we go. Consumer involvement has been, once again, we've heard about it, has been critical for the development of these trials. You know, we've had consumer involvement right from the start. They've been involved in the participant information sheets and consent forms, reviewed all of our advertising material, reviewed all study feasibility and design. They've creation of an information video, promotional materials, and have done media interviews about our trial. Again, we could not do it without the involvement of the Consumer and Community Reference Committee, and it's, again, you know, it's made our trial vastly better than it would have been without them. We've got a number of investigators, and we're coordinating here in Hobart, and our investigators in the various centres, and in particular in the Perron and centre. We've got Jenny here, who's been a major driver of getting this work up and going. We've got a trade table down there, which is where Vincent or Chigozi Nzigabi and Julie Sansom, who are running the trial in Hobart. They are doing all, you know, they enrol people, they do the treatments. We also have a couple of other PhD students you saw in there, which is Tram and Armin, who step in and help with the stimulation. I would particularly like to thank the Consumer and Community Education Committee. Particularly also like to thank Tash, who has not got to put a name on this slide. But Tash, we could not have done it without Tash's expertise in clinical trials. We'd also like to thank our Data Safety Monitoring Committee, which is critical for any trial. When when you're doing a trial, when you're doing any sort of research, you need someone to looking over your shoulder going, hang on a sec, is that really what you want to happen? And we've got a very good Data Safety Managing Committee. Sorry, this is a little bit frustrating. So the other thing, most important thing, is these trials are not cheap. It's really expensive to run a clinical trial, and we particularly like to thank the Irene Phelps Charitable Trust, who's funded both this project and also Nick's stem cell genetics project. I mean, we could not have done it without the Irene Phelps Charitable Trust. The Medical Research Future Fund, who's funded a lot of our research, has been unbelievably important in doing this. We also received funding for the TAURUS Run from Irene Phelps from the Royal Hobart Hospital Research Foundation. We also received it from MSA and from the Medical Research Society of Tasmania. So we have been able to attract funding from a wide range of sources. So I'd like to thank the event coordinators for, again, for asking us to talk about this. And if you're interested and would like to participate, please go and visit the trade table, speak to Julie or Vincent, and we're very happy to talk to you. Not everyone is going to be eligible, and it's not going to be for everyone, but really this is the closest you'll get to the cutting edge or the bleeding edge of technology. So thank you very much.